Hello friends, welcome to this class of T.Y.B.Sc Semester 1. I am Dr.Jayashri Bandal from Microbiology Department, KTHM College, Nashik. In this lecture, we are going to study the details of strain improvement, that is chapter 1. The unit 1 of this chapter includes objectives and selection of different types of mutants. In the previous lecture, we have discussed about the objectives of strain improvement and approaches of strain improvement. Among the two approaches for strain improvement, the first approach of modifying the genetic makeup has also been dealt with in the earlier lecture. In this lecture, we shall study the second approach of strain improvement, namely techniques for mutant isolation and the respective control mechanisms of the biochemical pathways which restrict the overproduction of the products. The techniques for mutant isolation again now become very important because the techniques for mutant isolation depend upon separating the high yielding strains from the average producers. So you have mutated the population, you have exposed the culture to a mutagenic agent, and from this culture now you need to separate the higher yielding strains because all the microorganisms in the culture that have been exposed will not mutate. So some of them will still remain as average producers, but those which will mutate, you have to select amongst them the high yielding strains. The changed character, that is, the characters which we need to improvise, should be taken as the selective factor for selecting the strains, improvised strains. The improvisation in the yields or the increased yields by the superior producers is due to all the substrate now being diverted to product formation. That is, our mutation program has brought about such a change in the microorganism that the substrate now is not shared for the production of two or maybe more different products. And so all the substrate now is being diverted to the product of our interest. This increases the amount of the product that is formed. The second reason why the microorganism now may become a superior producer or may become a high yielding strain is because it has lost the control mechanism or it has brought about a change in the control mechanism which earlier was limiting the amount of the metabolite that was synthesized. Limiting the amount of the metabolite that is was synthesized is the cell has its own regulation as to how much amount of the metabolite a microorganism will synthesize under particular conditions. And so, under natural conditions, a microorganism cannot overproduce a metabolite. Due to our mutation program, probably if there is a change in this control mechanism such that now this limit has been crossed or this limit is no more obeyed, then the more amount of the metabolized will be synthesized. So, knowledge of the biochemical pathway and its control will help us to hypothesize the blueprint of obtaining a desired mutant. So, if you know what are the factors that in the biochemical pathway that are controlling the amount of the metabolite that is synthesized, you can direct your mutation program to remove those controls. And this will help the microorganism to become a superior producer. So this such kind of a blueprint will enable the design of the isolation techniques for selection of the desired mutants. Thus, the knowledge of the biochemical pathway and its control are well studied for primary metabolites. Primary metabolites are metabolites that are required for the growth of the microorganism, like amino acids or vitamins or some other growth factors. So, their biochemical pathways have been well studied. And since the biochemical pathways and its control of the primary metabolites have been well studied, the strain improvement program for the primary metabolites can be easily designed because we have a well studied or a well very well known biochemical pathway and the control places. But the biochemical pathways for secondary metabolites, like antibiotics, are not readily available. Still, since a category like antibiotic has a large demand in the market, considerable progress in designing the procedures for the secondary metabolites have also been done. The regulation of the enzyme is operated at two levels. One, at the level of its synthesis, that is at the genetic level, which is called as feedback repression, and at the level of its activity, that is in the cytoplasm or wherever it is active, it is called as feedback inhibition. The metabolic pathways are either linear or branched. Why we need to study whether the metabolic pathways are linear or branched? Because the control mechanisms differ accordingly. Just to tell you about the feedback repression is, in the feedback repression, the end product of the pathway blocks the gene and it doesn't allow the gene to express, to produce the particular enzyme. And so it is called as feedback. Feedback because the product is going back to the gene, blocking the gene so that it will not express itself to produce the protein, that is the enzyme. Whereas in the feedback inhibition, the product binds to the enzyme and it blocks its substrate binding site such that the substrate cannot bind to it and cannot be converted to the product. So we can say that the feedback inhibition is an immediate level of stopping the excess production of the product. So let us exemplify the linear pathway in this way, where the substrate A is converted to an intermediate B, further converted to C, then to D, and then to Z. Different enzymes are operating for different stages. That is, enzyme A will convert A to B, enzyme small b will convert B to C, small c will convert C to D, and small d will convert D to Z. But the moment, that is, till the time this metabolite Z is required for some other activity in the cell, its concentration in the cytoplasm will remain minimum, because the moment it is produced, it is being diverted to the place where it is used in the further synthesis or some other metabolic pathway. But the time when the place where it is required is reduced, or its requirement for some other activities in the cell is reduced, it will start accumulating in the cell. And when it starts accumulating in the cell, it is present in the cell in enough amounts to go and bind to the enzyme, such that the enzyme can no more bind to its substrate, that is A, and thus the formation of B is stopped. No B, so no formation of C, no C, no formation of D, no D, no further production of Z. And so the product controls its own production. This is called as feedback inhibition. To exemplify this, we can take the example of the amino acid isoleucine. So threonine in a series of steps is converted to isoleucine. Isoleucine is an amino acid which is a part and parcel, which is a component of many different proteins, may it be the structural proteins or the enzymes. So till the production or the synthesis of those proteins is going on in the cell, there is a demand for isoleucine. So the isoleucine that is formed by this pathway will further be channeled to the synthesis of the different proteins. But when the protein synthesis of these proteins stops or is reduced, the requirement or the demand of isoleucine will reduce. And the cell has a very intelligent regulation that the moment the molecules of isoleucine start accumulating in the cytoplasm, it will go and bind to the enzyme which is converting threonine to alpha ketobutyrate, and it will stop the further production of isoleucine. That's just if you want to design a blueprint for isoleucine, that is, if isoleucine is your fermentation product, you need to know this, because isoleucine will never be overproduced by this microorganism. So unless and until you change this particular enzyme such that it no more binds to isoleucine, this is, we have unlocked or we have stopped the control mechanism, we have removed the control mechanism, then and then only the isoleucine will be overproduced by the microorganism. Amongst the branch pathways, the types of controls are feedback inhibition or cumulative feedback inhibition by isoenzymes or isofunctional enzymes, concerted feedback inhibition, and sequential feedback inhibition. Let us study each one by one. In the isofunctional enzymes, the feedback inhibition is seen in this way. Let us first see what are isofunctional enzymes. Isofunctional enzymes are two different enzymes showing the same function. So here you have an enzyme A and A prime. Both have the same function of converting the substrate A to the product B. Similarly, ahead in the pathway we have the enzyme C and C prime, which are involved, which are using the same substrate but converting them to different products. So here only A and A prime are the isoenzymes. Now, in the feedback inhibition by of the isoenzymes, enzymes A and C are under the feedback control of the product Y. You'll see here that A, if Y is overproduced, it will inhibit the activity of the enzyme C, as well as it will inhibit the activity of A. So if Y is overproduced, it is going to stop its production only. It will not affect the production of the other metabolite, because the other isoenzyme will still, which is not affected by Y, will still continue to convert A to B and B to C. And since this C prime enzyme is not affected by Y, the C prime enzyme will convert C to F, and Z will still remain producing till the concentration of Z in the cytoplasm does not exceed. The moment the cytoplasm of the concentration of Z in the cytoplasm exceeds, then it will go and inactivate the enzyme C prime, and it will also inactivate the enzyme A prime. So in the isofunctional enzymes, each product has the ability to regulate its own production, but it will not affect the production of the other product of that branched pathway. In the sequential feedback inhibition, as exemplified by this pathway, you'll see that if Y is overproduced, it will stop the conversion of C to D, but it will not stop the the conversion of A to B. If Z is overproduced, it will stop the conversion of C to F, which is further converted to Z. And if if Y is overproduced, and it stops the conversion of C to D, and Z is also overproduced, it will stop the conversion of C to F also. That is, under conditions when Y and Z, their amounts exceed in the cytoplasm, then only you will see excess amounts of C in the cytoplasm, which then will stop the conversion of A to B. So this is what is the sequential feedback inhibition. Now about the cumulative feedback inhibition. In the cumulative feedback inhibition, here you will see that overproduction of Y will affect only 30% activity of A, whereas overproduction of Z will affect 50% activity of A in converting A to B. But if Y and Z both are overproduced, then both will affect a higher percentage of activity, that is 65% activity of A, which will hinder the conversion of A to B and the further stages in this particular or in this particular type of feedback inhibition. In concerted feedback inhibition, you have enzyme A, which is not being controlled or not being affected by only Y. Neither is A being affected by only Z. Only excess production of Y will affect the enzyme C. Excessive amounts of Z will affect the enzyme C prime. And only if Y and Z both are in excess, the enzyme A will be affected, and then the production of Y and Z both will be stopped. To conclude with the regulatory mechanisms of the metabolic pathways or the enzymes, we have seen that the amount of metabolites synthesized by the microbes is regulated in many ways other than feedback inhibition also. All these regulatory pathways will allow the microorganisms to synthesize only the amount of metabolites that are required by the microorganism. So the microorganism is very choosy. It knows how to monitor the amount of metabolites, and so it will produce the metabolites in amounts only those that are required by the microorganisms. And this is very analogous to what we follow in our daily life. In the kitchen, we we synthesize, or we make only the amount of substances that can be consumed by the people in our house. We do not make more amounts. If we need, say, 5 chapatis every day, we don't go on making 50 chapatis. And the same, on this same principle, the microorganism also is working. Understanding the controls of metabolite production thus will enable us to construct a blueprint for mutagenesis and its isolation. That is, knowing these pathways and knowing these controls, we now know which controls need to be mutated, which part of the enzyme needs to be mutated, and then once you know which part has been mutated, you know which how to design a process for its isolation. Such mutants are called as postulated mutants. To summarize the key points that we have studied in this lecture, the strain improvement program improvises the microorganism in many aspects, or in the aspects which we desire. The productivity can be increased by modifying its genome, by mutagens, or by the recombinant DNA technology. It can be achieved by studying the control of metabolic pathways and the genetic map, which enables us to design the methods to isolate the mutant also. Such microbes are called as postulated mutants. So, dear students, I am very sure you have understood the basics of strain improvement, the other aspects of strain improvement, that is, how to isolate the mutants and to create the modular methods to isolate the mutants, and strain improvement by recombinant DNA technology, we shall be studying in the upcoming lectures, in the further lectures to follow. Thank you so much.